as we get old, we forget things. And therefore I thought I would write down what I would like to say. Yes. Professor Gautham, Professor Filament Raj, Dr. NLN, that's how we call it. Ladies and gentlemen, I want to begin by thanking the organization for arranging this important event. I also want to thank all the old students and friends of KMK who by their generous contribution and support made this possible for honoring my old friend and colleague, Dr. Karunakaran. I thank finally the team responsible for this meeting for giving me the honor to speak about my friend. Professor Karunakaran, C.B. Rajagopal and I joined the physics department as staff in June 1950. On the same day, we were appointed as demonstrators. I did not know them as I came here after serving other colleges for 4 years. But from the first day, we developed a deep friendship for each other. After a few months, one of the recently appointed lecturer left the department and Karunakaran was promoted as a lecturer, and he moved to room number 100 of Bishop Beaver Hall. KMK then was a very healthy young man full of life and vigor. He had a flair for teaching and learning new things. Room number 100 Bishop Beaver Hall became the meeting place for the self-styled intellectuals like T.N. Seshan, Raja Chellaya, who was the economic advisor to Manmohan Singh, Paul Daniel, and a sprinkling of staff preparing for the civil service examinations. They found their interaction with KMK intellectually stimulating. KMK was a complete teacher. He was a person of great intelligence, deep insight, and passionate about his work. And I would say the epitome of what it means to be a teacher and a friend. He prepared every lecture meticulously and wrote it down. He not only gave importance to the contents, but also the way it was delivered. He was eager to get the correct pronunciation of certain words by listening to the BBC radio broadcast. There was no TV those days. To him, every lecture was a performance. The remarkable thing was, he continued to give the same attention to his lectures even when he was not well. He could handle any subject with commendable mastery that was unique. In the early days when we joined the college, Dr. Tangaraj was our head of the department, and he never allowed any lecturer to take a subject for more than 3 years. And therefore, every 3 years there will be a rotation. And we used to have a competition who should get this. And therefore, all of us, except Karunakaran, we used to say, please allow me to take this class, and therefore we used to have our own competition. But Karunakaran will remain alone. He will take the subject which remained without anybody taking, because he was sure of handling any subject. He was confident of mastering any subject that was given to him. His lectures on quantum mechanics was the talk of the town. He had excellent command of the English language, and this took his physics teaching to another dimension. As a head of the department, he encouraged his staff to keep in touch with the latest in the subject and teaching methods by providing all the facilities needed to attend seminars, workshops, conferences, and summer school. On a personal note, we were good friends. See, we first worked as a demonstrator. In those days, there used to be what is called a demonstrator. Demonstrator means, tutors. In humanities, we were called tutors. In sciences, we were called demonstrators. We were helping only the lab work, and then sometimes some professors will give us to value papers. And all of us were asked to stay within the hall. I was allotted St. Thomas's Hall under Dr. Tangaraj, and C.B. Rajagopal was in Saliyur, and K.M. Karunakaran was in Bishop Eva. And therefore, most of us, K.M. Karunakaran was a good friend of mine, therefore I most of the time spent my time at Bishop Eva Hall. KMK and I used to buy new two-tall, as you know, I am talking about what things happened 60 years ago. We were very young and we wanted to look smart, and therefore we used to buy two-tall ties. Two-tall ties were the famous ties in those days. Dr. Boyd's, the then principal's rule was that we should either come with full Western dress or full Indian dress, not anything in between. And therefore we used to buy good two-tall ties, and our shoes, we wanted to make it shine, you know, as Dr. McPhail's shoes we used to see, our vice principal, his shoes were always shining, you know, you could easily see your face there. Therefore we also wanted to do the same thing, and Dr. Karnataka used to say, at least one end must be shining. Therefore we were staunch friends, and I found him a genuine, warm, enthusiastic, cheerful person with whom friendship was a highly valued commodity. His interest went beyond physics into a whole range of fine arts. He was very much interested in Carnatic music, cricket. He used to attend almost all the music performances during the music season, that is December. I was very fortunate to have had a friend and colleague like Dr. Karunakaran, and I am happy that the second endowment lecture is given by one of our illustrious sons of the physics department, and whose achievements have made us proud. Crystallography was also my favorite subject. I studied crystallography under the famous Professor G. N. Ramachandran. I am by this time you would have seen that I am a relic from the past. I am as much eager to listen to Dr. Gautham as you are all. So I stop here, and so that we will listen to Dr. Gautham. Thank you very much.
I deem it a great pleasure, and I am privileged to introduce the speaker of the day to you. Professor N. Gautham did his BSc Physics course at MCC. He completed it in the year 1975, after which he went on to MS University at Vadodara and completed his MSc in 1979. Then he went to Indian Institute of Science, Bangalore, where he finished his Ph.D. degree in the year 1983. After which he joined IISc Bangalore as a research associate, and he was there from 1983 to 85 for 2 years, after which he joined the Department of Crystallography and Biophysics at the University of Madras in the year 1985. And from then on, he is there in the department, and we all know him as actually I had an opportunity to work under him for a short period of time, and I learned a lot of things at that time. And he is presently professor at the University of Madras, and some of the awards he has got they are actually quite impressive. National Science Talent Scholarship in the year 1972 NCERT, then from the University of Baroda first rank in MSc, then from the Indian Institute of Science for the best thesis in physics and mathematics, the Martin Foster Gold Medal in the year 1983. Then from the DST, Department of Science and Technology, he got a fellowship in the year 1990. And above all, one of the important award is he was elected as a fellow of the National Academy of Sciences of India in the year 2007. That's an FNASc. And apart from that, he has a lot of research projects under him, which goes into several lakhs, and the total funds exceeds more than 2 crores. Sir has authored few books, of which 2 books are very important: Biophysics and then Bioinformatics, Databases and Algorithm, of which the Biophysics book is a prescribed textbook in several colleges, and he has 2 patents for MOLS, that is mutually orthogonal Latin squares. I had the privilege of studying about that. That comes under combinatorics. Then he has several peer-reviewed research papers, and several of his papers are have a higher number of citations. So with this impressive career, he has guided several students for their Ph.D. degree, and one of his current students is Mr. Sampal. Sir's daughter did her BSc UG at MCC, and now she is in the US. So actually we are happy to invite, actually when we initially thought of who should be the speaker for this year, when we invited him, he was a bit hesitant to do that because he was the first president of the Physics Alumni Society when we had our Physics Alumni meet in the year 2010. Then I was the president for a shorter period, and then now the mantle, we have actually asked Dr. Lakshminarayan to take upon the mantle and actually make us go and proceed further as the alumni. So with this introduction, actually I'm very happy to invite Sir. Thank you very much, sir.
Thank you, sir. Now I request our former and senior professor, presently the president of Physics Alumni Society and a very good friend of Professor Gautham, all subject teacher, our beloved Dr. N. Lakshminarayan, to come forward and present the memento to our chief guest. Thank you, sirs. Now I invite the chief guest, Professor N. Gautham, to deliver the endowment lecture, Crystals and X-rays Crystallography.
Yeah, let me begin by telling you how much of a privilege and honor I consider it to be invited to deliver this lecture, this endowment lecture, at the very same college where I spent so many three happy years, 4 happy years actually. We had a pre-university course at that time, so that was also a part of my stay here. So 4 happy years, a long time ago. Also it's a great privilege to be able to give this talk in the presence of Professor Arthur, who taught me all the electronics I ever learned. As I was telling him, this stood me in good stead when I went for the interview at IISc, but that's only the electronics. Whatever he taught me is the only electronics I know. I learned other things afterwards, I guess. I thank the office bearers and the committees of the Physics Alumni Society for inviting me here, for the second for considering it. I would like to think that they called me because I had something to say rather than just because I was, not just because I was a, a closed member and a founder member, let us say, of the Physics Alumni Society. Anyway, thank you very much. It was also opposite, I think, that because my own research career has been in the area of crystals and crystallography, and this year happens to be the, as you already know, happens to be the International Year of Crystallography as declared by the United Nations through their UNESCO organization. So, thank you everybody. Let me start by this picture it's not directly related to my talk, but it's related to the college. This was Professor Thangaraj, presumably, I don't know exactly, but presumably the teacher of Professor Karunakaran. I'm not very sure about that, but he was the head of the Department of Physics when this picture was taken, and earlier, that was in the 1950s. He's a very distinguished person himself, a Rhodes Scholar, and if you know what that means, he keeps company with people like Bill Clinton and Shashi Tharoor and so on and so forth, all of whom are Rhodes Scholars. So this was Professor Thangaraj. This was the founder of our own department, founder and first head of the department of our own department, the Department of Crystallography and Biophysics in the University of Madras, Professor G.N. Ramachandran. And this is one of the greatest physicists who ever lived, Professor Paul Dirac. Yes, he was visiting India for a year. It's a very interesting story why he visited India instead of going to the US where he was planning to go. But before that visit was due, he had gone to Russia. This was in the 1950s when there was a lot of anti-communism feeling in the US, and because he had gone to Russia, they did not give him a visa. So it's not only you and I who don't get visas. Professor Paul Dirac also doesn't get American visas sometimes. Presumably he also stood in the consulate, outside the gates of the consulate, and was told by some consul officer that no, I'm sorry, you cannot go to US. Anyway, he took advantage of that time, that sabbatical time that he had got from his university, to come to Bombay instead, to TIFR, where he spent a year. And during that time, he also made a visit to Chennai. So I don't know what they discussed and all that. There's no much record of this, but this photograph was there, and I would like to show that to you, especially because it has this Christian College connection. This, as I said, is the International Year of Crystallography, and this is the logo which we are supposed to display. This is the official logo of the IUCR, the International Union of Crystallography, which they have designed for this year. And this shows a diffraction pattern with the center spot being the 0 of 2014. And that is why I suppose I've been invited to talk to you about crystals and X-ray crystallography. Now, why was 2014 declared, one of the reasons why 2014 was declared the International Year of Crystallography is that in 1912 we obtained von Laue obtained the first X-ray diffraction image from a crystal. This was from copper sulfate, and you can see it's not a very nice image. I mean, any of you would be able to get a better image than this. But the first one, and it was in fact obtained by his students, not Laue himself. Laue was a theoretical physicist who was trying to use this idea of diffraction to prove actually whether X-rays, what are the wavelength of X-rays, and whether they were waves or particles and so on. So anyway, he was successful and he obtained the X-ray diffraction pattern. That was in 1912. Of course, they immediately, this was the very first pattern, but then immediately they were able to improve it and get very nice patterns, and you'll see some of those nice patterns later on. In 1913, immediately, William Lawrence Bragg and William Henry Bragg. William Lawrence Bragg was only about 19 or 20 years at that time. Yeah. So both of them proposed Bragg's law, and using that law, they also determined the first structure of sodium chloride. So they immediately recognized that they could work backwards from the diffraction pattern and obtain the detailed structure of what was inside the crystal. And this was the structure that they obtained. In 1914, the Nobel Committee recognized this important event and awarded a Nobel Prize to von Laue, and that's why in 1914, 100 years since then, is 2014, and 2014 is the International Year of Crystallography. And a year after that, the Nobel Prize was awarded to Henry Bragg and Lawrence Bragg, who was of course the youngest and still remains the youngest Nobel laureate ever. kind of Sachin Tendulkar of science, I suppose. Okay, and X-ray crystallography, I work in the application of X-ray crystallography to study biological structure, and increasingly this has become very important, how you understand biology using structures obtained by X-ray crystallography. But even though I mean, biology is one of the important applications, as you can see here, biology and medicine, pharmaceutical technology, and so on, that the application of X-ray crystallography extends over almost all the disciplines you can think of, including, I was surprised to see myself, planetary science. Some of the things that you study about other planets, etc., require X-ray crystallography. In fact, there is right now an X-ray diffraction equipment sitting on Mars in one of those planetary missions. So X-ray diffraction equipment has been sent to Mars to study the structure of the soil and minerals there. So this is, this is growing all the time. You want to study any structure, you have to use X-ray crystallography. This is about the only really method available now, though other methods do contribute, like nuclear magnetic resonance or electron microscopy. These do contribute somewhat to the study of structures. Ultimately, if you want to know the detailed three-dimensional structure of any molecule, you need to use X-ray crystallography. So this lecture will be more pedagogic. I will not be presenting my results so much. It will be more elementary, and some of you might be familiar with it. Please bear with me. And I designed this lecture for a wide audience, people from not only from physics but also from biology, people not only from from the master's degree or Ph.D. students, but also for undergraduate students. So you'll find some of it very simple. Now, what is a crystal? Okay, and why is X-ray crystallography so important? And what is X-ray crystallography? So that's what I hope you will be able to. By the end of the lecture, you'll have some idea about that. These questions, some idea about the answers to these questions. So a crystal is a solid material in which the atoms, or maybe molecules or ions, whatever is the repeating unit in the crystal, these units are arranged in an orderly repeating pattern which extends in all three dimensions in space. Right? So if you want to portray this in 2 dimensions, difficult to draw pictures in 3 dimensions, but if you want to portray this in 2 dimensions, this would be what we call normally a single crystal, where throughout the entire crystal you have all the units arranged perfectly in sync. You can also have this kind of material which is quite common. You have this polycrystalline stuff. This is where you have small regions of this material will form these microcrystals, and they would clump together to form some kind of a powder of the material. In fact, most metals, like the metal here or any metal that you can think of, most of them have got this kind of a structure. These structures, single crystals, also you must, there are some here, I suppose, in the hall. Mostly it will be jewelry or some of the components inside these electronic devices. They are all these kinds of single crystals. And the third state, in this series, if you go further and disrupt this orderliness, you get what is called amorphous material, which does not have any crystalline order. So these are crystals, and crystals are ubiquitous, as I said. You find them everywhere. But there are, of course, many things which are not crystals. For example, gases are not crystals, cannot ever be crystals. They do not have any translational order. Liquids are not crystals. All kinds of liquids. None of the liquids are crystals. And glass, this is a kind of a solid, and some other solids also are not crystals. Glass can look like a crystal, and in fact it is also called crystal by some commercial companies like Swarovski crystals you'll find. Yeah, these are not really crystals, they are glass, cut glass, and they are not crystals in the scientific definition of what we mean by X-ray crystallography. And this tells you some idea. There is no order in the way in which these silicon oxide atoms are arranged in the substance. So there are some very common crystals which you can find on your dining table, for example. Salt, sugar. You find these every day. You can go home and look at it, and they are crystalline. You may need to look at them under a microscope, but yes, they are crystalline. Quartz is not so common at home, but if you walk around in the fields, or I'm sure in MCC, if you walk around the ground, you'll be able to find some quartz crystals. They'll be just lying around. Sometimes they're mistaken for diamonds, but diamonds are different. Diamonds are also fairly common, I should think. Most of us have seen diamonds in jewelry, and they are also, these are some of the common crystals that you can see. And if you want to identify what is a crystal and what is not a crystal has sharp edges, it has flat sides, it has a regular shape, or it can be cut into making a very nice regular shape. It has a shiny surface usually, and it is usually transparent. Sometimes you have certain types of crystals inclusions which are not transparent, which are almost opaque, but usually crystals are transparent, even though they may be colored. And crystals will be symmetrical, which goes with this regular shape. So you can look at a crystal and identify that it is a crystal in most cases, but sometimes you are fooled because you look at glass and you think that that is a crystal. So the best way usually to check whether it's a crystal or not is to use X-ray diffraction. I'll come to this a little later, but let's go on and we'll talk about X-ray diffraction a little later, but let's continue with crystals, what makes up crystals and what we know about it. Of course, crystals were studied not only by Lovey or by Bragg. Long before that, people all over the world were looking at crystals. And this scientist, a French scientist, I don't know exactly how you pronounce his name, but I will just say René Jascoux, okay? More than 200 years ago, he recognized that the external regularity of the crystal, the externally regular shape in a crystal, actually reflects some of the regularity which is present inside the crystal. And then he came across the idea that you have these small units, which he called a unit cell, which can be arranged in a regular way in 3 dimensions to make up the crystal. So the actual repeating unit will simply be something that's present inside one of these unit cells, and that's repeated in 3 dimensions in various ways. The symmetry of the unit cell then is reflected in the external symmetry of the crystal. And in fact, for a long time, geologists, or even now, geologists use this to understand what is inside the crystal just by looking at the external shape and how it cuts. Okay, how you can cut it and what are called cleavage planes and so on. So that was recognized a long time ago. And if you make a mathematical abstraction, you can think of a crystal as built upon a lattice. This lattice, of course, doesn't really exist. It's just a mathematical abstraction. You have this units, these repeating units, which can be some molecules or atoms or something. And if you put a dot at the center of each and join them, you'll get a lattice like this. And this is just a mathematical abstraction which is very useful for us. So what is it that makes a crystal is actually the molecules or the atoms, but the way in which they arrange themselves can be thought of as this kind of a lattice. And now you can make an abstraction and talk about the symmetry of the lattice without in any way referring to what is actually present in the unit cell. So this unit cell is the smallest unit, and you can think of then as the crystal as being made up of the unit cell repeated in 3 dimensions. But the unit cell itself can contain sometimes just a single atom. Here you see 8 atoms, but there's only 1/8 of each atom in the unit cell. The unit cell, you can draw a unit cell by joining the centers of these spheres, and then you get 1/8 of an atom inside each unit cell, and therefore the total number of atoms in that unit cell is just one atom. Or you can have a few atoms. For example, sodium chloride has got a fairly large number of atoms of basically sodium and chlorine repeated, but if you look at the total number of atoms here, it will be about, I don't remember the exact number, but it will be about 3 or 4. So there will be 3 or 4 sodium and chlorine atoms inside this particular unit cell. So this whole thing is one unit cell. Even though you have these grids inside, actually this whole thing is one unit cell, and this is the actual arrangement of sodium and chlorine in crystals of sodium chloride. Or the contents of the unit cell can be a few tens of atoms. I'm not sure you can see this picture very clearly, but here you have a unit cell, and this molecule, which is a molecule of sucrose, is arranged. There are 4 or 5 molecules of sucrose arranged inside the unit cell here. You cannot have 5, 4 or maybe 8. Actually, here are 4, I think. 4 molecules of sucrose arranged inside each unit cell. So you can have, a few tens of atoms. Or sometimes when you go to much larger molecules like proteins, these are biological molecules, you can have as much as a few hundreds of atoms or even thousands of atoms inside each unit cell. So this is a very slightly complicated unit cell of hemoglobin. This is a protein that's found in all animals, and this is the structure that is determined by X-ray crystallography, but here we just talk about how you can have many atoms. Okay, so this is a detailed picture of the structure with each atom represented. It's very complicated, so you just make an abstraction and make it look like this and see that, yeah, it looks, these are the molecules that are present in the unit cell. So crystals can be made up from virtually any kind of material, provided the material is pure and homogeneous. You must have only one type of material in your sample, and then you can grow crystals. And these crystals will have these kinds of symmetries. We have, it has been, you can show mathematically as well as by experiment that there are certain types of symmetries which are possible in crystals, certain types of symmetries which are not possible. And if you look at the rotation axis, these 4 different types of rotation axis are possible whenever you have a lattice. Okay, whenever you have a 3-dimensional lattice, or even in fact a 2-dimensional lattice, whenever you have a translation symmetry which you need to impose upon the crystal, which every crystal you have to impose this translation symmetry, it has to have a unit cell which has to repeat in 3 dimensions translationally. So when you have that requirement, then it is only possible to have a 2-fold symmetry like a rectangle, or a 3-fold symmetry like a triangle, 4-fold symmetry like a square, or a 6-fold symmetry like a hexagon. Other types of symmetry like maybe pentagonal symmetry, 5-fold symmetry, or 7-fold, or any other rotational symmetry is not possible in a crystal. And this particular principle is unconsciously demonstrated by every day in front of our houses by the people who draw, the mostly women who draw the columns. And if you look at these columns as some of my students have done, you'll find that only these 4 different types of symmetries are possible in these columns. Specifically, I am referring to pulley columns, if you know what that means. Pulley column is when you place the dots and then draw the lines around the dots, like here. Okay. There are other types of rangolis where you can have other types of columns, but if you talk about pulley columns, then the symmetry that you find in these pulley columns are exactly the types of symmetry that you find in crystals. And this, in fact, was not my own observation. Dr. Gift Siromni and Dr. Rani Siromni, who were right here in Madras Christian College again about many years ago, in the Department of Statistics, I think, They have written about this and made some observations about this some time ago. So yes, and there is another type of symmetry. Apart from rotational symmetry, you can have these kinds of mirror symmetries, and these mirror symmetries are also present in in columns as much as in crystals. So you can have these 4 different types of rotational symmetries, and you can have this mirror symmetry, and you can have combinations of these along with the translation. So totally it has been shown that there are 230 different types of combinations that are possible, of these symmetries that are possible in crystals, and these 230 different types are called space groups. So each unit cell will belong, or each crystal will belong to a particular space groups, and one of the first things that you do when you have a crystal is to try to find out what the space group of the crystal is. So of course, before you can analyze a crystal, you need to grow the crystal. There are many different ways of growing crystals, depending on what type of crystal you are interested in. If you are interested in crystals made up of metals or things like that, then there are very like silicon crystals, for example, which is industrially very, very important. These crystals are grown using other techniques than what I'm describing here. But whenever you have something that will dissolve in a solution, in a solvent like water, or maybe even in an organic solvent, it's possible to use this evaporation technique to grow the crystal, which I've shown here. Normally what you can do is take this, I've demonstrated this, or I've shown this for the growth of protein crystals specifically. So what you need first of all is to obtain pure protein, and that is done by mostly the biologists, the biochemists. They give you pure proteins, and then you take that protein and dissolve it in the buffer, that is usually water plus some salts added to it, so that the protein retains its integrity. And then you prepare what we can call a crystallization cocktail with various chemicals, which will allow the protein to precipitate, but precipitate very gradually, very gently. Basically what you do is you reduce the solubility of the solution in which the protein is there, so that it comes out of solution gradually. Normally you put the precipitant plus the protein into a small drop here and hang that drop here above the reservoir which contains the precipitant, and then over a period of time hopefully you will get these kind of crystals in this drop. This has been a very successful technique used to grow crystals of well over 100,000 proteins, and the structures have been obtained, and this is a small sample of what these crystals look like. The coloring in these crystals, I must mention, is not inherent to the crystals, but you usually view these crystals under a polarizing microscope, and when you do that, because of certain properties of the crystals, the crystals give rise to different kinds of colors. So something like Newton's fringes, for example, these are something like Newton's fringes Newton's rings, that experiment that, I don't know whether you still do that, but this Newton's rings experiment that you may be doing in your BSc, that's the same phenomenon which gives rise to these kinds of fringes. Anyway, crystals grow in all different types. They grow in very unusual shapes, and in my own laboratory, Arun Richard and Madan Gopal were able to track the growth of some crystals of DNA, which initially grew as hexagonal plates, and over a period of 135 hours slowly dissolved in the center and became these kinds of ring crystals, which were stable for several months, or in fact years together. So this had a distinct hole in the middle. You could take it out, and then you could show that they were crystal by performing X-ray diffraction experiments and so on. This is about 10 microns or so. The diameter of this hexagonal ring is about 10 microns or so. Much smaller versions of such rings were observed right here in the physics department here by Rizwana and Dr. NLN. I've taken this without your permission, but I've given you credit. So, yeah, but these are also they are not from any biological material, they are from zinc oxide, and they were grown again using the same precipitation technique, and you get these rings. You also get these hexagons and you get these rings. I don't think they were able to track the formation of the rings from hexagons, but they were able to observe both in the same same method of preparation. But these crystals are much smaller than of the scale of half a micron. What you saw there was 10 microns. This is about half a micron or so, the diameter here. So yes, you can get crystals of different types and different shapes, and it requires a lot of effort. What I told you is only just one method which we use in our laboratory, but then there are several other methods which people use to grow crystals of all types. And in fact, all of you, any one of you can grow crystals, and to celebrate this International Year of Crystallography 2014, there is a crystal growing competition, especially for schools, and you need to go through a school. So now if you would be really eligible to participate, but you can tell this to other schools or maybe to your brothers and sisters if they know, if they'd be interested. So there is a worldwide competition open to all school children, and the winners will be those who most successfully convey their experiences, not necessarily the crystals themselves, but they have to grow the crystals and then describe what they did with the crystals. Either they can, they can make videos of it or write essays or whatever it is, and then you send it to the IUCR, and you'll be judged on that basis and awarded prizes. And this is the website for any one of you who is interested. More details about this is given here. Also some tutorials on how you can grow nice crystals at home, salt crystals, any kind of salt, sodium chloride, magnesium chloride. You can buy some salt like zinc sulfide or something and grow it. Okay, now once you have grown the crystal, what do you do with it? We have to study the crystals to find out whatever we want to do. Like we want to find out the symmetry, what's the kind of symmetry that's present inside, and or what is the structure, detailed structure of the molecules that make up the crystal. And these are some of the methods and apparatus that we use to study the crystals. Microscopes and goniometers. Goniometer is a device which you can fit the crystal on and study the reflection from the surface of the crystals. A goniometer is, these devices are used fairly commonly by geologists and mineralogists, and who are essentially interested in the morphology and in the external features of the crystals. Chemists are more interested in the compositions, and we use spectrometers. But if you want to know the atomic structure, detailed atomic structure, you have to use either X-ray diffraction or neutron diffraction. Both neutron diffraction requires large reactors, and also because the intensity of the neutron beam that you get is very small, therefore you need large crystals also. But X-ray diffraction is now, you can get X-ray diffraction equipment in your lab. You don't have to go to a huge 10,000 crore rupee reactor to get X-ray beams. So you'll see that yes, even X-ray diffraction equipment can sometimes be very expensive. But anyway, it's possible to get X-ray diffraction equipment in the lab, and you can use that to study, to generate X-rays and put the crystals on them, and to study the crystals. So what is it that you study using X-ray diffraction? Well, you can of course get details about the unit cell shape and size and the symmetry, what symmetry, whether there's got two-fold symmetry, four-fold symmetry, whatever. And ultimately, you can get the precise three-dimensional positions of all the atoms in the unit cell. That is to say, you can determine the molecular structure. The chemical structure, what atoms there are and how they are bonded to each other, usually you analyze that by, the chemists do that. They are able to find out what the chemistry is, how many atoms there are, what are these atoms, and how they are bonded to each other. But how they are positioned in three-dimensional space, how do they make up the structure, that is determined by X-ray crystallography. And these are, I already showed you this is the structure of sugar and this is the structure of hemoglobin. A little difficult to see each individual atom, you have to take my word for it. Here of course the red spheres here represent the oxygen atom, the black spheres represent the carbon atoms, the white spheres represent the hydrogen atoms. So this is how they are all represented. Once you have the coordinates, the precise three-dimensional position, you can use other computer programs to actually draw pictures like this. And now with graphics you can do all sorts of things. I'll show you some examples a little later. So X-ray diffraction is a phenomenon of, basically interference diffraction. We study this in school as, or maybe college, as two different phenomena. Actually they are the same phenomenon. And even though we call it diffraction, you would probably recognize it better if I call it interference. Like Young's double slit experiment, except that here the slits are made up of, each individual atom makes up one slit, and you don't just have two slits, you have all the number of atoms in the, each lattice point, for example, all the different lattice points make up the slits. So you shine a beam onto this crystal, which you can think of as a three-dimensional diffraction grating. Okay, so just like you get the diffraction pattern in one dimension in your lab, you can get it in 3 dimensions using X-rays from a crystal, because it's a 3-dimensional diffraction grating. And then you measure the X-ray pattern, you make various measurements, about the intensities and the positions of all the various diffraction spots or the reflections on the detector, and then you can work backwards and get what is the structure. So the crystal is a 3-dimensional diffraction grating, and since the spacing between these atoms, the space between the atoms, the size of an atom is approximately the same as the wavelength of X-rays, and therefore if you shine a beam of X-rays at a crystal, it will act like a diffraction grating and you will get a diffraction pattern. X-rays, if you shine a beam of X-rays as something much larger, you don't get a diffraction pattern. Likewise, if you shine, let us say, light on a crystal, you will not get a diffraction pattern, because visible light, the wavelength is much larger than the spacings between the atoms in a crystal. It has to be roughly the same order of magnitude. So X-rays that we use is of the wavelength of about 1 angstrom to 1.5 angstroms, which is the distance between, approximately the distance between two atoms, which will be about 3 angstroms or so. So the X-rays then produce a 3-dimensional diffraction pattern, and the diffraction pattern is then measured and analyzed to obtain the structure. So these are the different kinds of samples would give different types of diffraction patterns. If you have a single crystal, remember the first slide where I showed you, different types of crystals. If you have a single crystal, you would get a diffraction pattern with clearly defined spots. If you have a powder where you have lots of small crystals, you would not get a clearly defined spots, but you would get rings like this at different spaces. So you would get a bright ring, and then of intensity, and then you would get no intensity, and then again a ring, and so on. And you can take a cross section of this and then get the normal diffractogram that many of you may be familiar with. So this just tells you the intensity at different positions from the center, which is in fact an equivalent to the theta angle, the diffraction angle from the crystal. So theta and the intensity. And if you have fibers like this, okay, these are supposed to be fibers, I'm not sure you can see from there, but these are supposed to be fibers. If you have fibers where you have, this is also polycrystalline, but these polycrystalline units are aligned, okay, in one direction. They are not aligned in the other direction, they are all aligned along one direction. So you have these crystals stacked more or less on top of each other, but there is no 3-dimensional order. In the other 2 directions, there is no order. So when you have that kind of a situation, which is very frequent in biological materials, like if you take your hair or if you take a piece of your skin, these are all made up of fibers. In fact, this is a small piece of skin, a collagen fiber, and you'll get a picture which looks somewhat like this. Yeah, not somewhere in between this and this. Okay, not exactly distinct spots, but not rings either. So you can analyze these patterns and obtain details about the structures of the molecules or atoms that make up these powders or these fibers or these crystals. Ideally, you would like to have single crystals because that gives you the maximum amount of information, gives you very precise information about the three-dimensional position of every atom. But it's possible for small molecules and for certain types of fibers, it's possible to do the same thing using just this kind of diffraction patterns too. And this, I must say, I've said that this is almost the only way, because there are two other ways which I mentioned earlier, nuclear magnetic resonance and electron microscopy, but both of these have got large number of limitations. So if you consider, for example, the number of structures determined by X-ray crystallography and compare it with the number of structures determined by the other two methods, NMR and electron microscopy, it will be something like 10,000:1. So for every 10,000 structures obtained by X-ray crystallography, you may get one structure by NMR. But yes, the other structures, other methods are also possible, and therefore almost the only way to obtain the precise three-dimensional structure of any molecule is, first of all, you grow a crystal of the molecule. So you purify the molecule, get the molecule, grow a crystal of it, use X-ray crystal as an X-ray diffraction grating and measure the diffraction pattern. So you generate the diffraction pattern by shooting a beam of X-rays at the crystal, and then measure the diffraction pattern, and then analyze the diffraction pattern to obtain the structure. So this is another graphic representation of what you do. You start with a pure sample, you grow crystals, and then you put it in X-ray beam and you get a diffraction pattern which may look like this. This is for a complicated protein. You can see that there are hundreds of spots here, and in fact this is only one picture. You must, for each crystal you may want to take 100 such pictures. So you get about 100,000 of spots which you have to analyze. You basically measure. Obviously all this is done by computers nowadays. So you just put the crystal and then push a button and then go have a cup of coffee or go to sleep and wake up. So the data will be collected on the computer, and then once you have the data, you put it on a computer, and then you do what is called a structure solution and a refinement. You get, first of all, a function which tells you what is the electron density, which is the amount of electrons in each portion of your unit cell, and then you interpret this to determine the final three-dimensional structure, the positions of the atoms. The reason why you have only an electron density is because the X-rays interact with the electrons and not with the nuclei. So you can't see the nuclei using the X-ray diffraction. What you see are the electrons. The electrons are the ones that are doing the reflection of the X-rays. But that's good enough. If you are able to figure out where there are more electrons, obviously that's where the atom is. So you look at the electron density and then you do some interpretation based upon what you know about the molecule, whether it's a protein or what type of protein it is, etc. And then you can determine the three-dimensional structure. You can determine the positions of each atom and refine those to make sure that your data fits, your model that you create here fits with the data that you have. You refine it and finally you get a three-dimensional structure. Here again it's a different representation where each atom is represented as a sphere. So you have what is called a space-filling model where all the atoms are, there is a bunch of spheres. Once all you get are the coordinates. When you do this mathematical operation, you will get the coordinates, the numbers of the positions of the atoms in the molecule. It's up to you later on to use other computer programs to display this information in the way that's most convenient to you, and this is one of the ways to do it. Okay, there are two bottlenecks here. One is of course to grow the crystals. This is a very, very difficult job, especially for the more important molecules that we have now. So most of the recent Nobel Prizes that have been awarded in the field of X-ray crystallography, and I'll show you a list later on, are because they were able to purify certain types of very important molecules and grow crystals of that. And then of course they were also able to go through the entire procedure and obtain the three-dimensional structure. So there is one important bottleneck here, which is to grow the crystals, and the second bottleneck is to actually determine the structure. And why is this a bottleneck? Well, okay, I'll come to that, but one of the ways in which you can improve your data collection, okay, sometimes it's difficult to grow crystals, but you can grow, sometimes you grow very small crystals, and then you need very intense X-ray beams in order to study them, and those kinds of beams cannot be produced in the laboratory, but they can be produced in these kinds of large facilities called synchrotron facilities. Synchrotron is something that you all know, it's a particle accelerator. But as a byproduct of the particle accelerator, particle accelerators were originally built to study particle physics. But as a byproduct, people found that you get, because the particles are going around in a circle, you have electromagnetic radiation coming out at the so-called bremsstrahlung, if you know that, if you have heard that name before. That electromagnetic radiation comes out in a tangent as the particles spin round and round. And you can use that electromagnetic radiation. It can be of all different wavelengths, and you can tune it to get X-ray wavelengths, and these are very powerful beams. If you want to use the facility only for particle physics, then you take precautions to shut out that and make sure that things are safe. But if you want to use it for studying, use that X-ray radiation, you put in beams, and you put beam holes or beam lines and draw out those beams, and then you can use that for X-ray crystallography or for other kinds of work. And these kinds of facilities are now available all over the world, including one which was recently inaugurated in India and Indore. And this is a picture of one such source in France. And the diameter of this would be roughly around half a kilometer or so. The bigger they are, the more powerful they are. This would be about half a kilometer. And what you see, actually there will be a ring inside, and there will be lots of experimental beam lines, as they are called, which are coming out from them. So you can use these kinds of facilities, produce very intense X-rays, and use that to study the structures of molecules. And this has revolutionized the study of biological molecules, especially because now you are no longer have to grow very big crystals. You can take very small crystals and put them on one such beamline and get the data, solve the structure. There is still the other problem, structure solution, what is called the phase problem in X-ray crystallography. The mathematics of this is simply that if you have the electron density, then the X-ray diffraction experiment tells you that this electron density can be determined as a Fourier transform of the structure factors. What are these structure factors? These structure factors are nothing but the diffraction intensities and the phase associated with each diffraction intensity. So this thing that is drawn in red are called, written in red are called the structure factors. And if you know the structure factors, you just have to perform a Fourier transform and obtain the electron density. And then once you have the electron density, you can interpret it and refine it and determine the positions of the atoms, and so on. But the problem is that the X-ray diffraction experiment gives you pictures like this. From this, you can determine the structure factor amplitudes, but you cannot determine this value of phi, which is phase. There is no way in which you can measure it. So, if you are able to determine this value of phi, which is the phase, then you can use this Fourier transform expression straight away to obtain the three-dimensional structure. But so the phase, this is called the phase problem in X-ray crystallography, and again several Nobel Prizes have been awarded to for people who have contributed to ways to solve it. There are of course methods to solve it, but these are very tedious. This is just to tell you that it's not a straightforward job to go from the X-ray diffraction data to the structure because of this phase problem in X-ray crystallography. There are methods of overcoming it, and once you overcome it, you will be able to obtain the three-dimensional structure. So, yes, we do all this. Why do we have to do all this? Well, I already gave you a list right in the beginning, which told you about the applications of X-ray crystallography determining the structures. Now, this is more support for the idea that you can use X-ray crystallography, and it's important, from Max Perutz, who won the Nobel Prize for the structure of hemoglobin. When somebody asked him why we need to know the structure, he said why water boils at 100 degrees and methane at minus 161 degrees. Why blood is red and grass is green. Why diamond is hard and wax is soft. Why glaciers flow and iron gets hard when you hammer it. How muscles contract, how sunlight makes plants grow, and how living organisms have been able to evolve into ever more complex forms. The answers to all these problems have come from structural analysis, in particular structural analysis using X-ray crystallography. So you can see a wide variety of different fields or areas of research is covered right here. And all these answers to all these questions can come from X-ray crystallography. A well-known example of how the structure explains the properties and function comes when you compare the structure of graphite to the structure of diamond. Graphite is soft. Graphite and diamond are both made up of carbon, exactly the same kind of carbon. Yeah, carbon atoms are carbon atoms. But graphite is soft, but diamond is hard. Graphite is among the softest of substances known, and diamond, of course, as you well know, is the hardest substance. Once you have analyzed the structure, in fact, you analyze the structure using X-ray crystallography, you find that graphite has got this kind of structure where even though the carbon atoms are bonded like this strongly within the plane, between the planes the bonding is very weak, and therefore the graphite will simply disintegrate if you just touch it or push it. Whereas in diamond, every carbon atom is bonded to the other carbon atom very strongly, and there is no different types of bonds. All the bonds between the carbon atoms are very strong, and therefore diamond is very hard. So this kind of analysis can be obtained only when or this kind of explanation can be obtained only when you do X-ray crystallography and determine the three-dimensional structure. So this is a very simple example, but you can find such examples in various other fields. In biology specifically, why do you want to study protein structure? You want to understand biological function, to understand and prevent disease, to design and manufacture drugs, to design better agricultural techniques. All these things, and these are kind of applications, but these are, understand biological function is a more fundamental scientific reason why we study protein structure. So in the last part of my lecture, I will just go through some slides which will tell you some of the structures which, a few structures which are important, which I think specifically protein structures which are important. This was one of the first structures of protein that were determined by Professor G.N. Ramachandran, whom you saw as a very young man in the very first slide. He is thought to be I should write G.N. Ramachandran FRS here, but he is thought to be the probably the preeminent scientist of independent India. Okay, you had scientists too, C.V. Raman, etc. before that, but in independent India, maybe he is certainly the preeminent physicist or biophysicist in independent India. So that's he determined the structure of collagen using fiber X-ray diffraction. Collagen is obtained as fibers, and you need to put these fibers into an X-ray diffraction machine, and you get a fiber diffraction pattern, and you can use that, analyze that to obtain the three-dimensional structure of collagen. That was one of the first structures, but this is a more recent structure. Just to tell you how detailed you can get, you can actually see each individual hydrogen atom. This is a part of a large protein, and it's possible using X-ray diffraction to get very detailed picture of each individual hydrogen atom. This is supposed to be a methyl group with a carbon and 3 hydrogens, and you can actually see the positions of the hydrogen atoms. One single electron, and you are among, please let me know, the molecular weight is 35,000, so there will be roughly around 100,000 electrons in the molecule, and you are able to see the position of each electron. So this is the kind of resolution that you can obtain. This is not always possible, but sometimes, if you're lucky, it is possible to get this kind of resolution. And these are some very important structures, and these are all Nobel Prize winning structures. This is the structure of hemoglobin, which is again a very important protein. This is the structure of insulin, which is important for me especially because I'm diabetic. So yeah, so this is structure which is determined again by Dorothy Hodgkin. Both these, incidentally, these names which I show you are all visited our department at some time or the other. So we have some kind of a family relationship with many of these people. They are now no more. Both Max Perutz and Dorothy Hodgkin have passed away, as most of the other people have shown you. Aaron Klug, he determined the structure of a virus particle. So this is a protein, and then he showed that this protein molecule is arranged like this in a kind of a spiral, and that is what makes a virus, a particular type of virus. Other types of viruses are slightly different. And this is the structure of a very complicated molecule called a ribosome. And Venki Ramakrishnan, along with a couple of other people, won the Nobel Prize for this particular structure in 2009. So you can see we can use modern day diffraction technique, computer graphics to make these kinds of displays. You can do all sorts of things. In fact, you show structures by making movies and zooming in and zooming out and all that. Otherwise, it's very difficult to explain what exactly you're seeing. But ultimately, these are all done by computer programs after you get the structure, which is just the coordinates. And these are some colorful structures from my own laboratory. I will not really explain them too much. It's just different types of DNA structures. I work in DNA crystallography, and these are some structures from my own lab. So just to impress you a little bit more, if you're not impressed already about my X-ray crystallography, you'll see that even in 2013, that's last year, the Nobel Prize was awarded to 3 people for some, actually these are, they didn't actually solve any structures, but they determined, they had developed new techniques which will help us to solve protein structures. Okay, so that's to Karplus, Levitt and Marshall, and then regularly you see, okay, this is only in the last 20 years, but since the advent of X-ray crystallography, a total of 48 scientists have been awarded the Nobel Prize for something related to X-ray crystallography. And this is just a small selection over the last 20 years. Okay, much of them is for biological molecules. For example, this is for biological molecules, 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 this is for biology. Some of them are for, maybe material science or chemistry, graphene, fullerenes, and some of them are for methods. Okay. So, like for example, better neutron diffraction from, these people got it for neutron diffraction from crystals. Sharpa got it for better detectors to make, he made these so-called area detectors or multi-wire proportional counters as they call it, which are also used in particle physics, but they are very useful in X-ray crystallography. So I will end this with a quotation again from P.M. Dirac. Dirac was in the first slide, and it's correct that I should have him also in the last slide maybe. 1947, he said that the main object of physical science is not the provision of pictures, but is the formulation of laws governing phenomena and the application of these laws to discovery of new phenomena. And that's what he said. He's a theoretical physicist, he's quite happy with equations. But most of us are not, especially biologists and many physicists are not very happy. We like to see things, we like to imagine things. And that kind of seeing things, seeing molecules, seeing atoms, how they are arranged, is done mainly by X-ray crystallography. So thank you for your attention. I would also like to thank all these students who contributed to some of the work that I showed here. I should also, I should have already included the name of Monica, one of my students who is here in the audience now. I'm sorry Monica, but I didn't think of it. Yeah, she's also contributed to some of the structures that I showed. Thank you.
Thank you, sir. The talk was very interesting with more pictures and details. You have answered us most of the questions. What is it? Why we should learn about it? And how is it done? And when things were done? Thank you so much, sir. Students, now the floor is open for discussions and interactions.
when the virus is non-cellular, isn't it really difficult to do the X-ray diffraction in a virus? Can you please explain?
Yeah, the virus is extracted from the cell, or even if it is not in the cell, it's extracted from wherever it is. It's purified so that you get only the virus particle and it's crystallized. You grow crystals of the virus and then you do the rest of it. Take the crystal, put an X-ray beam and generate the structure, diffraction pattern, etc.
Isn't the virus inert outside the body, outside the host?
It's inert, but we can always show that if you take that crystal of the, that virus crystal and dissolve it, let us say, in some solution and inject it back into whatever it infects, the infection goes on. So the virus is live. Live in the sense of it still has its function. See, a virus is an organism which is not, we don't know whether it is a living organism or not. It's not a cell, but it's something more than just a protein. So we don't know what it is, but we can study the structure. You don't really have to know what it is. But it's not as easy as I make it sound. It's a very difficult job to extract the virus, purify it, get the crystals, and then get the structure. But people have done it. And
neutrons will get embedded to the crystal.
no. The neutrons, if they are powerful enough neutrons, basically the wave particle duality, if you have neutrons of the specific energy that you require, they will act exactly like X-rays. In the sense they diffract. You can get a diffraction pattern from neutrons. They interact with the nuclei. Neutrons will interact with the nuclei, so the equations become somewhat different. But essentially it's a diffraction pattern again, and essentially you have to do a Fourier transform from the diffraction pattern to determine the positions of the nuclei right now, rather than the positions of the electrons. So you don't get an electron density, but you get the nuclear positions of the nuclei, which is good enough. I mean, you can use that for determining the structure. The trouble with neutron diffraction is that, as I told you, you require the flux that you get, the intensity of neutron beam that you get is very weak even in the reactors, whereas X-rays are many-fold more intense. So you need to grow large crystals, and that's not always possible for the kind of molecules that you have. There are those problems, safety problems, hazards.